Hello, and a very good afternoon to everyone, and welcome back once again to the second session of the day, one day national seminar on quality assurance, essential, validation and IPR guidelines, organized by Adarsh Shikshan Prasarak Mandal, K.T. Patil College of Pharmacy, Dharashiv. Now we will move towards our second session, which focuses on another vital pillar of quality assurance, that is validation. Friends, validation is the backbone of pharmaceutical industry as it ensures that every process consistently delivers products of the required quality. It is not just a regulatory formality but a scientific approach to build confidence that systems and processes are reliable, reproducible and under control. In simple terms, validation ensures that what we are doing is correct, consistent and reliable. In today's highly regulated environment, validation acts as a bridge between theoretical quality and practical performance, ensuring that what is designed in theory is effectively achieved in practice. With this understanding, we now invite our respected speaker to share deeper insights into the important topic. I would like to give my warmest welcome to the speaker of second session, respected Dr. Jayaprakash Samsetti sir, professor at YB Chavan College of Pharmacy, Chhatrapati Sambhajinagar. Welcome, sir. But before moving to the actual session, I would like to give a brief introduction of Dr. Samsetti sir. Dr. Jayaprakash Samsetti sir is presently working as professor at YB Chavan College of Pharmacy, Chhatrapati Sambhajinagar. He has completed his B.Pharm from YB College Pharmacy, Aurangabad, M.Pharm from Mumbai University, and doctoral degree from Dr. Bamu University, Chhatrapati Sambhajinagar. He has around 25 years of professional experience. He is approved PhD guide of Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, and 6 students are working under him. He has 10 patents, 2 published and 8 granted to his credits, and has published 298 research papers in international journals with a cumulative impact factor more than 650 in Thomson Reuters and H index of 46. He authored 6 books and also written 8 book chapters in books published by Nature Springer, CRC Press, Elsewhere and Bentham. His research work has received 7,200 citations. He is a reviewer for more than 100 international journals. 5 of his research papers have been ranked by Science Direct as hottest 25 articles. He is a recipient of Young Scientist Award from DST. He has successfully completed projects received from DST, UGC and AICT and SARB. Total cost 1.5 crore. He has received a Best Paper Award from Elsewhere in 2015. He also has received Young Pharmacy Teacher Award from Maharashtra State APTI in 2015. He has also received V Life Best Publication Award in 2014. He has received PD SETI Award from Best Research Paper in 2009. He has guided more than 49 M.Pharm students. He worked as a member board of studies of pharmacy at Shivaji University, Kolhapur. He is working as international panel member of Project Evaluation Committee of National Science Center, Poland, since 2015 and evaluated number of projects. He also worked as a brand ambassador for Bentham Science Publisher between 2017 and 2019. He has received prestigious Padma Manmohan Sharma Science and Technology Award 2023 by Marathi Vidyan Parishad. He has been included in top 2% researchers list published by researcher of Stanford University for the year 2020 and 2025 consecutively. His major area of research includes design and synthesis of bioactive molecules, application of quality by design in drug development, synthesis of polymers and their application in gene delivery system, development and validation of analytical methods for drug and pharmaceutical dosage form. There is a lot more to tell about Dr. Jayaprakash Samsetti sir, and we have very short time period, but we have huge uh, reading for the CV. So, but we are truly honored to have such experienced professional amongst us who will guide us through both the scientific and regulatory perspective of this important topic. So, thank you, sir, for accepting our invitation, sir. Before we begin, a few instructions for our online audience. You may post your questions in the chat box. Question answer session will be conducted at the end of this lecture. So now I would like to hand over the mic towards the Samshetty sir and would like to request to start his session. Please.
Thank you, madam. Am I audible?
Yes, yes, sir. Yes.
Yes. Okay, okay. So, thank you very much for the introduction given by Angar madam. So, basically, the topic that I have selected for today's discussion, that is about ICHQ14 and how this ICHQ14 will be the future era as far as pharmaceutical analysis is concerned. So, pharma industry and the pharma regulations, they are in the transformation mode. And what changes are going to take place in the future pharma industry as far as pharmaceutical analysis is concerned, since analysis it is an integral part of drug development. So we cannot escape from this particular drug development process. As such, we have just it requires some time. I will just share the presentation. Once it will be thorough, I will see. Yes, is it visible now,
Yes, sir. Yes.
madam?
Yes,
Yes,
sir.
yes,
Yes.
yes.
It is
Now you can
visible.
see this particular slide. So basically, this is a very big topic. Practically covering that entire topic in 1 hour, it is quite difficult. But still, within this particular time span, I will try to cover what are the important aspects as far as quality by design is concerned, and how this particular quality by design uh, in the framework of analytical pharmaceutical analysis needs to be taken into shape as far as the future of pharmaceutical analysis is concerned. Now, before going to that particular details about this ICHQ 14, which I am going to talk about, we should know what exactly this ICH is. Now, this is the supreme body that governs the guidelines related to the quality as far as pharmaceutical drug development is concerned. And that's why this particular body plays an important role in streamlining the quality guidelines, in implementing the quality guidelines and harmonization of all these particular guidelines throughout all sectors of drug development, and in case of pharma industry as well as in any other aspects where the pharmaceutical research is going on. Now, in this particular presentation, what I am going to cover, I will just give an overview. Basically, I am going to speak on what exactly the quality by design is, why we have adopted this concept, then how the changes in the regulatory perspectives have taken place in order to acquire this particular concept in the pharmaceutical development. And we are now at the stage where the pharmaceutical analysis, which is an integral part of drug development, that needs to be fit in the framework of quality by design. And certain guidelines have been introduced in this ICHQ 14. How this ICHQ 14 differs from the existing ICH guidelines, and how what are the different historical perspectives as far as introduction of ICHQ 14 is concerned. Now, to start with, what exactly the quality by design is? It is a particular science. That particular science, it is adapted by the pharmaceutical sector. This science, it is addressing some of the issues that has been faced by the pharmaceutical sector since long time, and with that particular hope, it has been adopted through the introduction of ICH guideline 8, 9, 10. Initially, later on, different ICH guidelines have been introduced in order to expand the scope of this particular ICH guidelines, and introduce the concepts of quality by design that can be applied for the different sectors of drug development. Now, there are many definitions that has been given in as far as QBD is concerned. The definition which has been given in ICH Q2, Q8, sorry, it is a systematic approach to the development that begins with the predefined objectives and emphasize on the product and process understanding. What quality by design is? It is a process, or it is a particular science-based and risk-based approach that is totally depending on understanding the process. If you want to gain the success in any process, understanding that process is very important, and that's why quality by design, it is a science-based and risk-based approach that is totally depending on how importantly or how you are gaining the control on the process and how you understood the process. Quality by design, it identifies the characteristics that are critical to the quality from the perspective of the patient. It translates them into the attributes, and it establishes how these critical process parameters can be varied. As I mentioned just now, QBD, it is not a new science. It is the science which is existing, which has been exclusively explored by different science like chemical engineering. We have adapted it to address some of the issues that we were facing as far as pharma development is concerned. And to some extent, we are successful as far as addressing some of the issues that we were facing 10 to 15 years back. Now, what exactly it is totally depending on understanding the process. If you want to get the success in the process, we need to understand it. What are the different parameters that affect the performance of that process that we are identifying? We are naming them as critical quality attributes. We are separating them. We are gaining the focus on these particular attributes. Gaining the focus on these particular attributes and gaining the control on the process ultimately helps in success of that any process. And that is the basic theory as far as quality by design is concerned. So again, this is the difference between the current approach which we were following, that is quality by testing, and QBD approach, that is quality by design. So every approach, every new approach that we are adopting to address certain issues, it may have some advantages, and it is definitely helpful for addressing some of the issues that we were facing. So there were certain issues associated with the approach that is known as quality by testing. So we are judging the quality at the end of that entire process, and we are taking the decision about that entire batch. So there were, as far as QBT, that is quality by testing, is concerned, it is a frozen process, means there was less scope for variation after the application. Any variation in after the application was considered as a change. But on the contrary, QBD, it is a flexible process. We can make the changes if provided that changes should be within the design space. What is the design space? It is nothing but the multidimensional combination of input variables that are affecting the performance of that particular method. Then more focus has been given in QBT, that is on the reproducibility, whereas in case of QBD approach, the focus has been given on the robustness. Robustness parameter, which is the key validation parameter that has been mentioned in ICH-Q2, it has been widely explored in QBD. That QBD can be applied for all the stages of drug development, including API synthesis as well as analysis. Much literature is available on how we can implement that QBD for different stages of drug development, but specific guidelines have been given in this ICH-Q 14 that we are discussing today. Again, any concept, it is based on particular thought. This particular concept, it has been introduced by a quality expert, Joseph Girard. He suggests that quality cannot be tested into the products, but it has to be built inside by the design. So we cannot judge the quality by testing at the end, but we need to develop that particular product of desired quality. In other words, we have to define what are the expectations as far as quality is concerned from the product at the start. Accordingly, we need to work on that particular product so that the initially defined objectives that we framed at the start can be achieved. So QBD, it is a concept which is based on predefined objectives, and predefined objective, it is considered as a core of concept of quality by design. Now again, what makes pharmaceutical regulators to implement this concept in the pharma sector? I just now mentioned quality by design, it is not a new concept. We have adopted this particular concept. The implementation, it is not sudden. It took many years because pharma regulators were facing many problems as far as harmonization of the different guidelines are concerned. Different regulators, different manufacturers working in the different parts of the world, they were following the different guidelines of their own countries, and no specific guidelines were available which are common for all. So the genesis of utilization of QBD concept in the pharma sector, it has been in one of the event that took in at around 2000, where FDA has received more than 5,000 supplements as far as applications to the NDAs and ANDAs are concerned. So supplement, it is an additional application which is submitted to the regulatory body, which which is asking for permission for some of the changes that you have initially applied to. So why such type of changes were occurring? That was the issue. That was the major question in front of regulators. And regulators framed one committee, and they have decided why more supplements are applied for a particular application. So the reason was less focus on the process, more focus on the chemistry. And considering this, it has been decided to give more focus on the process rather than the chemistry. Chemistry is also important, but along with the chemistry of the processes involved, we need to focus on the exact process. What are the parameters that affect the performance of the process? What are the parameters that may hamper the objective that we set at the start? And all these particular parameters need to be segregated. We need to give again the control on that entire parameters. And with that thought, QBD has to be implemented in the pharma sector, that has been decided. But since it is a very big industry, implementation, it should not be in 1 year, because it requires certain preparations, and they have decided to bring all the pharmaceutical manufacturers on one common platform. With that view, process analytical technology PAT that has been implemented, it is first step. In 2005, US FDA has asked some of the firms to submit their application in the form of QBR, that is known as question based review. And ICH Q8 and Q9 and Q10, they have been implemented at around 2005-6. That is the first step for implementation of quality by design. And some of the basics which are involved in the concept of quality by design has been framed out in ICH Q8 and 9 and 10. The initial definition that has been given by the regulators in some of the documents published by the regulators is QBD, it is nothing but ICH Q8, Q8, 9 and Q10. And since 2013, for generic products, QBD has been made mandatory by US FDA. Slowly, all the sectors of pharmaceutical development have been covered. Now, regulators are up to a particular point where how the analysis can be reframed in the environment of quality by design. Now, as far as ICH is concerned, parallelly, regulators, they have drafted the guidelines in terms of ICH. The expectations from the implementation of quality by design in the pharmaceutical development has been framed in the ICH guideline. Initial definition, as I just now mentioned, it is QBD is nothing but ICH Q8, Q9 and Q10. But later on, it has been observed that in all these three ICH guidelines, no specific information about the nature of API, the process of synthesis of API, or the manufacturing aspect of API has not been covered. Then ICH Q11 has been implemented, incorporating the methodological aspects, incorporating the manufacturing tools or variables that plays an important role in the synthesis of API. Then in case of ICH Q12, some important information and considerations expected by the regulators have been given as far as product life cycle management is concerned. In ICH Q13, continuous manufacturing of the drug substance and drug product, so that has been mentioned. And in ICH Q14, much information about how analytical method development will be reframed in the QBD framework as far as drug development is concerned. But now, implementation of any one of the ICH guideline, it is not a single day task, or it is not a single steps task. Usually regulators used to publish one concept paper before say implementing any guideline. They will give the concept paper. In the concept paper, they will give the rough outline, what should be the picture of this ICH guideline. They will ask you the suggestions regarding the incorporation of many other points that are missing. As a pharmaceutical analyst, as a pharmaceutical professional, everybody can contribute to this particular ICH guidelines. Considering the suggestions given by all the professionals, the rough draft of that particular guideline will be published. As far as ICH Q14 is concerned, it has required, say, more than 5 or 6 years, because concept paper has been published in 2018, and because of pandemic, that particular concept paper could not take a shape of final draft, and later on, in 2021, again the concept paper has been published, and they have asked for the suggestions. Considering the suggestions by the professionals, these particular guidelines related to ICH 214 has been draft, and in 2023, final adoption of this particular ICH guideline was there. Before that, in 2022 November, October, so these rough guidelines or crude guidelines have been published, and finally the final guidelines have been published in November 23. Now, why this particular ICH guidelines Q14 have been implemented? Everybody was asking about, no specific guidelines are available as far as pharmaceutical analysis is concerned. So we all the pharmaceutical professionals, they were focusing on the guidelines that has been given, that is ICH Q2. ICH Q2 speaks about the methodological aspects of different analytical method developments and their validation. But the framework of QBD is totally relying on only one validation parameter, that is robustness. Now, it is very much essential to explore that robustness for incorporation in the ICH Q14. They don't want to disturb the primary guidelines which have been given in the ICH Q2. And later on, they have drafted the guidelines in such a way that they have divided that entire guidelines into two subparts. They have considered the primary approach as a minimal approach, and some of the components they have incorporated in another type of approach that is known as enhanced approach. I am coming to all the aspects of this ICH Q14 in the next slides. So again, how this particular definition has been turned into, the initial definition was 8 plus 9 plus 10. With some suggestions, ICH Q11 has been introduced, considering the requirement for API synthesis and their role. Then pharmaceutical life cycle management plays an important role. ICH Q12 has been incorporated. Continuous manufacturing of the drug substance and drug product, ICH Q13 has been incorporated. And at the end, ICH Q14 has been incorporated. Now, the question comes, what next? Usually, introduction of different ICH guidelines as per the need of time. ICH is updating themselves. Even if some of the guidelines have been implemented based on some suggestions received from the pharma professionals who are actually practicing these guidelines, they give the suggestion to ICH, and these guidelines have been amended from time to time. So as a pharma professional, as a researcher working in the field, we must always be in touch with ICH website, and we need to see how amendments are continuously taking place. Now, what are the different elements of quality by design? As I mentioned, quality by design, it is a concept which is based on predefined objectives. Means what we are expecting from any process, we have to define at the start. Accordingly, we have to work on the entire process, and we need to achieve the objectives. So defining an objective, it is the first step. Determination of critical quality attributes. Now, what are critical quality attributes? We will come into later on. Once you identify the critical quality attributes, we have to go for the risk assessment. Means we will identify what are the different steps which are considered as a more risky, means which will affect the performance of the process. So if we are working on any process, the performance of the process, it all depends on many parameters, many factors, or many, we can say, different parameters. It has been named as in critical quality attributes. So these attributes plays an important role. Controlling these attributes plays an important role in success of the entire process. So risk assessment, development of experimental design, designing and implementation of control strategy, and continuous improvement, these are the key important segments. And all these segments have been considered in ICH 214 as far as analytical method development is concerned. Now, if you see the literature, Quality by design can be successfully applied for variety of analytical methods. These methods are routinely used in the drug development process, which includes LCMS, UPLC, and many more. I have enlisted all the analytical tools where QBD can be successfully applied. Much literature is available on this particular data. Basically, though HPLC is widely used tool for analysis of pharmaceuticals, but many more other analytical tools are required for identification, for assigning the impurities, for quantification, and many more activities that are related with the qualitative and quantitative aspects of drug development process. And that's why all these particular analytical tools have been covered under QBD framework, means QBD has been successfully applied for all the analytical tools that I have mentioned. Now, what are the advantages? Actually, regulators are suggesting that yes, we need to adopt this particular ICH-Q14. What ICH-Q14 is, it is nothing but an advancement of ICH-Q2R1 that I am going to explain later on. So regulators are suggesting it is a mandation on all the pharmaceutical developers to implement it. So what are the different benefits? The robustness parameter, to summarize this slide, I can say that robustness parameter, it has been widely explored in this analytical quality by design. As I mentioned, in ICH-Q2R1, all the validation parameters, including robustness, has been gained much focus. But here, the robustness has gained much more focus, and the exploration of that particular process, as far as analytical method development is concerned, is majorly through the exploration of robustness. So, these are different advantages I have enlisted. It provides space for invention of new techniques. It helps for understanding of the method. It provides the regulatory compliance, even if you have many questions about its implementation. But as the regulators are asking for the implementation, we have to adapt it. Now, before implementation, before drafting the guidelines of implementation as far as ICH-Q14 is concerned, so on what parameter these particular guidelines have been implemented. So for that, I have some background as far as HPLC method development is concerned, that we have already mentioned in one of the book that we have published in 2014. These guidelines are published in 2023. But when we are focusing on the different aspects of, say, pharmaceutical development, much information was not available about how analytical method development can be fitted into the quality by design concept. But whatever the literature was available, that literature suggests that there were two approaches that has been suggested by the two key researchers or team of researchers working in the two major international firms, GSK. That is Oct et al. He has suggested one approach that is known as MDS approach. MDS approach, it is nothing but a method development strategy. And that particular method development strategy, it has been represented like this. Now, this was the concept which has been initially given by Oct et al. And this particular concept, which has been initially mentioned in one of the journal that has been published in Journal of Pharmaceutical Sciences. And if you see the basic thought behind this particular concept, again, the basic thought is hello, am I audible?
Yes, sir. Yes, you're
Yes,
audible.
yes. Okay. So, if you see this method development strategy, this particular method development strategy, it has been initially published in 2011, as I just now mentioned in the previous slide. So again, the basis is same. What is the objective of analytical method? You want what you want to measure, and how the results of your analysis are affecting on your entire product, or what is the intended use of method, then what type of information you want to gather from that particular analytical method. So we are naming it as an method goal or objective, which is analogous to the setting the objectives as mentioned in the basic concepts of quality by design. Once you define what you expect from analysis, the second job comes: which analytical tool will be helpful for giving the answer of question I am expecting. That is known as method scouting and evaluation. So suppose the answer is yes, HPLC will give the answer. HPLC analysis will help us to give the answer of question that I'm expecting from it. Then you need to develop the method under QBD framework. And once you select the method, then what are the different parameters that affect the performance of analysis? Performance of your method means performance of your analysis. So for that, you will list out the different parameters, and these particular parameters we are calling it as an method parameters. Once you identify different parameters, next job comes: which parameters are more risky, which parameters are less risky. So method selection and risk assessment. Risk assessment, identifying the risk mitigation, minimization of the risk involved in the process, and ultimately we are getting the control. So once you identify the risk involved in the process, we will go to how will you gain the control on the process. Gaining the control on the process, it is totally depending on focus on the risky parameters that you have identified. So once you gain the control, ultimately we can achieve the objective that you set at the start without any deviations. And once you develop the method, in 2011, before existence of these particular guidelines, validation parameters were also incorporated in the analytical method development, because remaining four steps that we are carrying out, it involves only exploration of robustness. So different analysts suggest that yes, no doubt you have explored only robustness. Validation is required. So whatever the method you optimize, go for validation of remaining validation parameters. So validation, it was one of the important tool that was explored. But again, with that time, with some suggestions given by different researchers, it has been decided that yes, validation is essential, but validation should not be the method objective. Previously, we were developing the method, and without validation, we cannot proceed. But in QBD framework, what they are suggesting that validation should be an additional tool. It should not be a method objective. And this particular thought has been incorporated in ICH 14. And we are coming to that particular part. Another approach which has been suggested in 2013, I am talking about how researchers were coming, we can say, with the new ideas, and how these particular ideas have been helpful for implementation or drafting of the guidelines. So whatever the research data available, whatever the literature data available as far as implementation of quality by design is concerned, these two suggestions, one is by Ocht et al. And second one, it is by George Reed, who is working as a analytical head in the Pfizer. This particular paper has been published in Journal of Pharmaceutical Sciences in 2013, and he has suggested AQBD workflow. AQBD workflow means analytical quality by design workflow. Again, the thought is same. The thought or basic thought of quality by design has been applied for analytical method development. And he has given a particular chart or workflow. According to him, again, if you see this particular workflow, it is depending on knowledge management, method life cycle, and continuous improvement. Continuous improvement, it is an integral part of any of the process. Based on the feedback received from the previous work, we need to improve the process next time so that we can get the better success. Now, if you see this analytical quality by design workflow, here he has suggested that analytical target profile is a term which is synonymous to quality target profile. What exactly analytical target profile is? Analytical target profile, it is a term which will explain about what are the requirements from the method. If you see the previous particular method development strategy approach, ATP is synonymous to the objectives or method goal. Then this particular ATP, once you set, yes, I want to develop a particular method, analytical method, or I want to develop a HPLC method with a number of theoretical plates, say more than 2000. Then once you set that particular objective, you want to work on it. Then you will identify what are the different parameters, what are the different process parameters that will affect the performance of my method. So I will list out, say, 5, 6, 7, 8 parameters. Now, all 5, 6, 7, 8 parameters, they are not significant much. Now, for that, I need to categorize them. I need to go for the risk assessment. I need to segregate them based on the nature of risk. And for that, I am going for the DOE. Once I identify them, which are more risky, I want to reduce the risk involved in it. Complete removal of risk, it is practically impossible. So we have to bring the situation where the risk has to be reduced to the minimum. And for that, we are going for de-risking with the help of DOEs. And once you go with the de-risking process, ultimately it will come with a space that is known as design space. And for analytical method development, they have used the term MODR, that is known as Method Operable Design Region. What it is exactly? So here, as far as HPLC method development is concerned, we are giving the wavelength not with the fixed value, but we are giving the wavelength in the range. We are giving, say, composition of mobile phase not with the exact value, but within the range. We are giving the column on temperature not with the exact figures, but within the range. So we can make the variation and we can make the changes in that particular range. And if you want to submit that application to the regulatory bodies for the for approval, and later on if you want to make the changes, then we need not go for additional supplement. So supplement is needed only when if it is out of design space or out of MODR. So this particular important aspect has been suggested in 2013 and 2011, and these two workflows or these two approaches suggested by the different researchers, it forms the basis of ICH Q14. Okay. What are the contents? The important contents of ICH Q14. It includes, it has some objectives. Any of the ICH guideline, it is always implemented with certain objectives. So what type of objectives they have set? What are the expected benefits that regulators expect that yes, with the introduction of these particular ICH guidelines, pharma professionals, they will get benefited with this. What is the scope? Then they have given two types of approaches, minimal approach and enhanced approach. What this minimal approach is, we will see. For that, we should have some knowledge of ICH Q2, which we were traditionally using. Then analytical procedure life cycle, analytical target profile, knowledge and risk management, evaluation of robustness parameters, control strategy, life cycle management, all these tools that has been mentioned in ICHQ 8, 9, 10, 11, 12, 30, they forms the basis of ICHQ 14. So ICH Q14, it is a blend of primarily ICH Q2 R1 and then ICH Q8, 9, 10, 11, 12 and 30. So as such, if you see the objectives, so this particular guideline, it will serve as a primary source of information for any analytical chemist who want to work on any of the analysis which is involved in the drug development process, where two types of approaches have been suggested. The minimal approach that has been given in ICH Q2, it is mandatory, and some of the approaches or elements that has been suggested in ICH Q8 and Q9 can be additionally used. That is the suggestion given by ICH in ICH Q14. So traditional method development is mandatory, but use of few components which have been mentioned in ICH 8, 9 and 10, they are also mandatory for implementation of ICH Q14. Now what are the expected benefits? harmonization of scientific approach, understanding of analytical procedure is increased, then enabling the preventive measures, reducing the amount of efforts. So the trials which we used to conduct in a traditional way of analytical method development, that can be reduced in case of this uh, AQBD, that is analytical quality by design methods. So what is the scope? It applies to all types of procedures that are new as well as revised. It can also be applied to other analytical procedures that has following the risk-based approach. So since it is a science-based and risk-based approach, the basic thought given by quality by design in ICH 8 and 9, it remains same, considering analytical method development as one process. The different parameters required for success of analytical method development needs to be listed out. The risky parameters needs to be segregated. Focus is given on all these risky parameters and control strategy has been planned. The method will be developed and some of the components of validations which have other than say robustness, we can go for an additional checkbox tool. And that is the basis of ICHQ 14. Now, what is minimal approach and what is enhanced approach? As far as minimal approach is concerned, this is the approach which is given in ICHQ2. Identifying the attributes needs to be tested. Then selecting appropriate technology and related instrumentation. Conducting appropriate development studies. Defining analytical procedure description. As far as enhanced approach is concerned, evaluation of sample properties, defining ATP, which is synonymous to QTP or QTPP, then conducting risk assessment and evaluating prior knowledge, conducting multivariate experiments, defining analytical procedure control strategy, and defining life cycle change management plan. So basically, this ICHQ14, it is a blend of ICHQ2 along with the basic concepts that has been given in ICH 8, 9, 10, and 11. Now, if you see analytical procedure life cycle that has been given in again ICHQ 14, prior knowledge means what? Prior knowledge, it is knowledge about the product. Prior knowledge, it is nothing but the knowledge about the route of synthesis of that. If I am working on say API, from where that particular drug substance we have procured, what was the route of synthesis that they followed, what are the process related impurities, what type of solvent has been used, what was the residual solvent, what is its proportion, and whether that particular residual solvent, it is from which class. Again, there are three classes as far as impurities potential of solvents is concerned. So it all comes under prior knowledge. Now, even if you have procured the API from authorized vendor, the route of synthesis plays an important role. API can be synthesized by a number of routes. The types of impurities, types of process-related impurities may vary if the route of synthesis is different. The types of impurities introduced inside the final API, even if they are in the acceptable range, because the coming era and the future era will be the era of genotoxic impurities. The prior knowledge of the product is very important as far as analytical procedure development is concerned. Now, ICH M7, it speaks about how this particular analytical methodology plays an important role as far as genotoxic impurities are concerned. So prior knowledge is very important. That will help us to identify the analytical tool. That will help us to identify which analytical tool will helpful for giving the answers of the questions that we are getting from that particular analysis. So prior knowledge, selecting the proper technology. Once you select it, then you have to go with the different particular parameters that we need to set up. Then you have to identify the different parameters. We are calling them as an risky parameters. Segregation of parameters that you have identified as far as any analysis is concerned, it is very much important. There are different tools available in ICHQ9 that we need to refer. And once you identify, yes, these are the parameters which are more risky. These are the parameters which are less risky. Focusing on the more risky parameters is very important. Why it is very important? Because the variations in the process, we want to get the success in the process. So for that, we need to focus on more risky parameters. And once you will gain the focus, ultimately you will get the success in the process. That is the basis of quality by design. So defining analytical target profile, it is again one of the important aspect and one of the important helpful aspect for selection of the technology. Once you select the technology, you will go with the risk assessment, and then you will identify the parameters. You will go with the segregation of the parameters, identify control planning the control strategy. Once you plan the control strategy, you will develop the method. You will validate the developed method for the parameters other than robustness. Why not robustness? Because robustness, it is one of the parameters that we have widely explored in AQBD process. That's why we need not go for the robustness again. So this is all about analytical procedure life cycle. Now, prior knowledge is very important, as I mentioned just now, not only with the in the case of API, but it is also important in case of formulations. There are many formulation excipients that may have interaction with the API at different conditions. The impurities which are reported by the we can say initial inventor, or which are given as a information by any vendor who is supplying to for the formulation. Besides this, some of the impurities that has not yet been detected by any of the analytical method can also be a part of your analysis if you could identify such impurities. And many aspects which were not explored by the traditional analytical method development has been explored by using this analytical QBD. And different researchers, they are working on how this will be helpful for identification of unidentified impurities. If X drug is there and there are 8 impurities which are reported with that particular X drug, so QBD has helped out to identify ninth impurity which has not yet been identified. That is the most important aspect. This is because of focus on the robustness, the more exploration of robustness in addition to other method development parameters. So again, ATP, these are some of the definitions. Analytical target profile. Again, ATP, it may be anything which is synonymous to quality target profile. What we expect, it is nothing but a definition of objective that we set. A prospective summary of performance characteristics describing the intended purpose and anticipated performance criteria of analytical method. So in simplest word, what I am expecting from analysis, that is nothing but an ATP. Yes, for example, I want to develop HPLC method with number of theoretical plates more than 2000. So this is an ATP for my method I want to develop. Another ATP, it could be, I want to develop a simultaneous estimation method for component A and B, whose resolution is more than 4 minutes. So this is an ATP for my analysis. So expectation from the analytical method, it has been incorporated in analytical target profile. So what target I want to achieve? ATP may be related with your process. ATP may be related with, I can tell you another type of ATP, identification of all reported 5 impurities for API. That may be ATP for any API for which there are more say more impurities reported. So there are number of literature reports, there are number of research papers that has been published, which will use such type of analytical target profiles. So analytical target profile, it is nothing but the expectation from that particular analysis on which we are working on. Now, knowledge and risk management in the analytical procedure development and continual improvement. Now, knowledge management, as I just now mentioned, prior knowledge, it is very important. Prior knowledge में क्या आता है कि from where the drug has been procured, what was the route of synthesis, how it has been crystallized at this last step. Now, highlighting more on knowledge management is basically the drug can be synthesized by using different routes. Synthetically, the drug can be synthesized in say 3 steps, to some extent 5 steps, or more than 5 steps also. Now, impurities are introduced at every stage. If the impurities are not removed at particular stage, and if impure intermediate is taken for the next step, that impurity remains in the final product as an process related impurity. Sometimes, even if no impurity has been introduced up to last stage or up to n-1 stage, and if I will go with, say, some chemical transformation at the end stage, the impurity may be introduced at the end stage. If I will take care of all the stages of drug synthesis and I am crystallizing the final drug product using a suitable solvent, the solvent after crystallization may get trapped inside. We are calling it as a residual solvent. Now that residual solvent is also serving as an impurity and it may interfere with your analysis. So knowledge management includes all. So what is the route of synthesis? What type of solvent has been used? Now, in the era of genotoxic impurities are gaining much more importance in last say one or two decades. So we were talking about impurities in ICH-Q3, but controlling impurity is not sufficient. What are the impurities are there? The concentration of impurity plays an important role. Their genotoxicity plays an important role. If these particular impurities, even if they are not harmful today, but later on, if they are producing some effect on the genetic material of the humans, then such type of impurities are not acceptable as far as pharmaceutical drug development is concerned. Industry people, they are using the term suspect for all such type of genotoxic impurities. So a list of more than 10,000 chemicals has been published. These particular 10,000 chemicals, they may be solvents, they may be some intermediates, they may be some of the fragments which are formed as an intermediate in the organic synthesis. More than 20,000 chemicals are under evaluation, the genotoxic potential of which will be, the data of which will be available very soon. So practically drug development, day by day, it is becoming tedious. So we need to focus on all the aspects. Since regulators are concerning with it, we need to also focus on all these aspects. So knowledge management involves prior knowledge about the product, prior knowledge about the nature of product, prior knowledge about the source from where you have procured that particular starting material or API or excipients also plays an important role. Again, quality risk management, the tools that has been described in ICH-Q9 that will be helpful for segregation, identification of risk, risk identification and risk minimization. These are the two important tools as far as quality risk management is concerned. And these two tools, different types of tools are available. I am not going in much detail about all the tools available. Evaluation of robustness and parameter ranges of analytical procedures. So the second diagram that I shown just now, that particular AQBD workflow which has been given by Reed et al., which has been published in 2013. It is one of the important segment of ICH-Q14. See, any ICH guideline, it cannot be drafted by 4 or 5 people sitting in one room. So these particular guidelines have been primarily drafted by some experts in that particular area. The draft becomes open for all for suggestions. Initially, the concept paper is given to all. Some suggestions will be asked based on the concept paper. Rough guidelines will be published. Once suggestions will be received for rough guidelines, final guidelines will be drafted. So as I mentioned in initial slides, where MODR, which is a key component of AQBD workflow, which has been given by Reed et al. In 2013, it is forming one of the basis of this particular ICH-Q14. Then control strategy, again in line with ICH-Q12, in line with ICH-Q13, controls in line with the primary guidelines which has been given with ICH-Q9. The control strategy needs to be planned, and we have to give how this particular segments or particular parameters that we identified, they are more risky or less risky. Then life cycle management and post-approval changes of analytical procedures. Here, initially the risk assessment will be performed. Then more risky parameters, they have been treated with a different paradigm, and less risky parameters, they have been reported as such. The focus will be given on more risky parameters, and they have been treated as a special case where more focus will be given on more risky parameters. And sometimes during life cycle of that particular product, if certain situations are arise where we need to make certain changes in the original analytical method development, this ICH guideline provides a scope for such type of changes. So this is all about life cycle management and post approval changes. Then multivariate development of multivariate analytical procedures. Again, focus is on the robustness and life cycle of a product. Then real-time release testing, some special considerations have been given. So it is based on an appropriate combination of one or more process measurements. Then RT-RT approach and product CQAs, they play an important role in proper justification. Then consideration for sample and sample interface are provided, and the impact on specifications is very much important. Now, I have given an overview in 1 hour discussion. Practically, it is very difficult to cover all the aspects because whatever the approach has been developed as far as basic guidelines are concerned. So it is a combination of say 3-4 ICH guidelines. Basically, they have not changed the framework which has been given in ICH Q2, but some of the components they were missing in ICH Q2 have been added. Along with that, these particular added components and the traditional ICH Q2 guidelines, they have kept as a minimal approach, and they have suggested to incorporate some of the enhanced approach where you should take the help of ICH 8, 9, and 10, specifically 8, 9, where more focus will be given on the risk analysis, that is risk identification and risk minimization. So combination of these two approaches will be future as far as analytical method development is concerned. So elements of QBD has been incorporated. Minimal approach that has been mentioned in ICH Q2 is mandatory. Enhanced approach, which includes some of the components of ICH 8, 9, 10, have been suggested as an additional support. And combinedly, we can develop the analytical method incorporating all these segments, and we can say that the method will be developed as per ICH Q14. Now, last important aspect, whether it will be a real roadmap for the analytical method development or not. The answer of this particular question, it will be available not immediately, because whenever any guidelines will be implemented, unless and until these guidelines will be thoroughly practiced by the industrial professionals, and limitation of these particular guidelines unless and until they will be focused. Some of the amendments which are needed in these particular guidelines, unless and until these amendments will be available, we cannot say that it is a real roadmap for analytical method development. So this is all about an AQBD. It may be a roadmap, new roadmap for analytical quality by design. So these are some of the important publications related with this, where this book we have written in 2014, where we have just mentioned about ICH 8, 9, and 10. 11 was just introduced. Later on, 11 has been introduced, ICHQ 12 has been introduced, 13 has been introduced. We have written one chapter in this particular book where analytical method development, what should be the future, where we have mentioned about the 2 cycles that I have suggested in this particular presentation. And fortunately, the second approach, which has been given by John Reed from Pfizer, it is forming a part of ICHQ 14. So with this, I am going to conclude this particular session. I am again thankful to Principal Joshi sir for the invitation, Coordinator Biradar madam, and our respected Principal sir for giving me an opportunity to interact with you.
Thank you, sir. Am I audible,
Yes.
sir?
Yes.
So thank you, sir, for insightful session. Your valuable inputs have helped us to understand the tricky concept of QBD, which is used in quality and promoting innovation. So as the session was very informative and looking into chat box, our participants has benefited by the guidance they have got from you. While the presentation was going on, the participants were asking some questions regarding today's topic. We would like to pick up some questions from you. Shall we start the question answer session, sir?
Yes, yes.
Okay, sir. The first question is, can AI help in identifying critical quality attributes and critical process parameters more effectively within QBD framework?
Basically, in general, I will answer this particular question. Everywhere, AI will not be helpful for. AI will give some of the answers based on its available data that has been fitted into that particular related to that particular question. But as far as CQAs are concerned, in analytical framework, the CQAs are depending on many parameters. So even if you will put the prompt for AI about what should be the CQAs for X drug, or if you are analyzing that particular X component or analyte using a particular analytical tool, AI will use some of the tools that are general for all types of drugs or analytical methods. Specifically, CQAs are the properties of two important aspects. First one, it is the property of analyte, and it is the property of analytical method that you are following. So ultimately, if you see number of literature reports as far as AI is concerned, so majority times it may mislead you rather than giving the correct answers.
Okay. So, sir, the next question is, according to QBD, how do you ensure that the defined design space remains valid during the scale-up technique and commercial manufacturing?
Yes, yes. Question, please repeat it.
According to QBD, how do we ensure that the defined design space remains valid during scale-up and commercial manufacturing?
Yes, yes, yes. Uh, Success of implementation of quality by design to the commercial scale. That is a question I think
Yes, yes,
you are asking about.
yes, sir, yes, sir.
So, if you ask me, no such documents are available openly. But I used to discuss with many of my industry colleagues. They are able to handle many of the situations where number of failed projects of last decades, means last 10 years, such projects were failed because of many reasons, and they got some of the answers using the QBD approach. So the success rate, if you ask me about how this particular design space implementation and QBD framework will be helpful for commercial implementation. So in some cases, where industry was failing to address the issues that they were facing for many years, some of the issues has been resolved. So I can tell you one example of one drug that has been synthesized, and as far as API synthesis is concerned, there is a tremendous competition among the different companies as far as cost of API is concerned. So basically, when we introduce any product in the market, if that cost of the product is 1 lakh per kg, now we cannot guarantee that that particular product will be available in the market for 1 lakh kg for many years. I cannot sell it. Another competitor will come and he or she will sell the product at 60,000 per kg. So tremendous competition is there as far as API development is concerned. But one of the product where impurity was the biggest challenge, and that unknown impurity was making a problem as far as bringing that particular product in the market. But use the help of uh, quality by design approach, where chemometry, it is one of the important aspect where API developers, they used to apply it. That chemometry has helped it. With the combination of chemometry and QBD, they have designed the synthetic route where the level of impurities has been reduced to close to zero, means within the acceptable limit, and they bring that particular molecule in the market at very cheap cost. And the company has progressed, say, 3 times more than their usual progress. So in some cases it is helpful, in some of the cases it is not helpful. So again, answer of this particular question is not, we can say, we cannot give that yes, it is 100% successful. but it has helpful for the industry for addressing many issues, and many issues are not yet been addressed. Next one.
Next question is from our final year student. That is QBD. For QBD, which ICH guideline should pharma student read first? That is Q8, Q9, or Q10?
For QBD,
Yes.
basically what I suggest is for the B Farm students, I cannot say that you should start with 8, 9, 10, but I suggest that you should start with ICH guideline 1, because some of the aspects of impurity has been given in ICH Q3, again Q4, Q5, Q6. So for graduate students, the learning sequence should be from ICH Q1. But if you jump directly to ICH Q8, many of the primary terms that has been used in the ICH 8, 9, 10, okay, you will not get that particular primary terms. So gain the primary insight on the basics of guidelines, quality guidelines. You have to start with ICH Q1, and once you thorough with the basic aspects of quality guidelines, then you can start from 8, 9, and 10. As such, QBD concept has been introduced in ICH Q8, 9, and 10. 8, 9, and 10 are important.
Okay. Now it's my personal question, sir. As a
Yes.
researcher, what common mistakes should we avoid while performing validation studies in our project?
While performing,
Validation studies in our project.
what? Can
Uh,
you repeat, madam, question?
as a researcher, what common mistake should we avoid while performing validation studies? While performing validation studies, sir.
While performing validation study, what?
Yes, in our project, sir, research project, sir.
Validation, if you are developing the analytical method in QBD framework,
Yes, sir.
validation is not mandatory. It is an additional checkbox tool. Means, since we are exploring one robustness parameter thoroughly, we can go for validation other than robustness.
And the
Means
last question is,
we have to optimize the analytical method development. The optimized method development we can validate, except robustness means we can go for other parameters of validation.
and the last question is, sir, as we are living in this AI tools and AI automation system, so how do the emerging technologies like automation or AI influence the QBD and validation practices today?
See, number of tools are available. AI as such, it will be helpful, but to majority extent it will be helpful, but to some extent it may mislead, as I mentioned just now. It is again a database tool. So whatever the data we have fed it, means we humans are providing the data, and based on the data they are interpreting it. So every time we should not rely on it, but we can take the help in to some extent for getting some of the important aspects. But as initially one question has been asked, so can we get the idea about CQAs using say AI? So for such type of approaches, we need to work on the nature of API or nature of analyte, and then analytical tool which we are considering. So answer of this question is every time we should not rely on AI.
Okay. So it looks like we have covered all of our questions. So thank you participants for your active response, and thank you sir for resolving all the queries of the participants. So now I would like to extend my sincere gratitude to our respected speaker Dr. Jayprakash Samshetty sir for giving their valuable time and sharing their insightful session with all of us. So I would like to thank you from management as well as our institution. So to Jayaprakash Samshetty sir for giving this informative session to all of us. Thank you, sir. Thank you very much for joining.
Thank you, madam. Thank you very
Okay.
much.
Thank you, sir. So I would like to again thank you for the organizing committee, the convener of this seminar, Dr. Amol Joshi sir, and the coordinator of this seminar, Dr. Birazar ma'am, and organizing committee for their dedicated efforts for successfully conducting this seminar. A special thanks to all the participants of our YouTube audience and for your active presence and engagement. We hope today's seminar has strengthened your understanding of validation and IPR and will support you in all academic and professional learning. Thank you all the audience, and thank you, sir. Thank you once again.